person on Zoom, and there's like 40 people on the Zoom. So she said, you can't have the side talks and other things like that.
Hi, Ron.
Hey,
哎，
哎
喂，
喂喂，
哎
yeah,
yeah, 小莹
long time no see.
long
Long
time.
yeah. 嗯
That's great. Yeah.
yeah, our group remote.
Yeah, we wait 1 more minute. So lots of people like log in currently now. It jump over in 160 now. Yeah.
Oh, what'd you say? How many more minutes?
1 more minute.
One more. Okay.
Lots of people log in.
last minute.
Yeah, always. Yeah, I think we have like 170 or even gross number now. Let's start. So I give a introduction first. So good morning to audience in China and good evening to people in North America. It's a really great privilege and honor to introduce Dr. Ron Evans from Salk Institute. Everyone know that. So Ron is a well-known scientist who cloned the first nuclear receptor, glucocorticoid receptor GR, and pioneered the nuclear receptor field. He identified multiple nuclear receptors there, including thyroid receptor, vitamin A, D receptor. After discovery, RR heterodimer with other nuclear receptors, he promoted the field, clone even more nuclear receptors. Ron's career path, reflect the progress. Literacy this one, yeah, okay. Made in the field. Um, Ron's career path reflects the progress made in the field of metabolism, endocrinology, cancer biology, circadianism. Start from molecular cloning, identify new genes in 1980s, then characterize molecular mechanism of those nuclear receptors in 1970s. That's the reason I include Dr. Hou Yingao as a panelist. That's the kind of earlier generation of Ron's trainees. Then determine the physiology functions in 2000s, and his work continued for new field actually included recently. There's a lot of requests for the recording. And include our bioacids metabolism, and include a new concept of this called biome. Actually, that's we have also over more than 600 papers that are not covered in this introduction. Instead, I would introduce our panelist. I mentioned Hong Ying in early 1990s. Actually, same year, joined Ron's lab as Peter Thomas. Xiaoyong in the middle 2000, he have published a famous paper about circadian rhythm of all nuclear receptors. And Fu Ting, I just mentioned, working on just recently independent 3 years ago, maybe, or 4 years ago, focused on bioacid research. Um, if we include like Li Ming, Zhong Wei, and other former trainees in Ron's lab, that's easier to turn Ron Evans' lab meeting. So we also invited Dr. Wen Donghuang, who trained with David Moore, working on lots of nuclear receptors and bioacids. Also, we have Lei Ying, who trained with Mitch Lazar. She worked on NR1D1, another nuclear receptor, also known as reverb. Ron also make a lot of contribution in this circadian rhythm field and also PPR alpha now. I'm confident we will have a lot of highly inspiring discussion after Ron's lecture. So last but not least, I also would like to extend our appreciation to Nova Nordisk for their long-term support to this seminar series. So without further ado, please join me to welcome Dr. Roy Evans for today's seminar. The title, I think, is Nuclear Receptor Overcoming Insulin Resistance, Cancer, and the Obesity Epidemics. Ron, the stage is yours. Thanks a lot for joining us.
Okay, thank you very much. And it's really a pleasure to be able to be part of the Metabolic HD Talk series. And it's so great to see so many of my colleagues in attendance here. And then they're going to be asking questions like I always ask them questions. And so it's really a pleasure. And I'll be talking a little bit about nuclear receptors a little bit, and then delve into some aspects of, I should use the, can you see the little pointer here?
Yes, I can see your mouse. Yeah, I can see it. Yeah, it's good.
It's not so big, but okay. So I'm gonna, I'll be talking obviously about nuclear receptors because they're amazing, and with the undertone of overcoming insulin resistance, which is one of the major challenges in around the world in general, and then cancer and the obesity epidemic. So these are kind of intersecting components, and I think that it's going to be a very this is some of the new advances I think are going to be very important. Right. Did that go at all?
Good.
Okay, now we're back on track. Okay, so basically, you guys, and I think most people know something about the nuclear receptor superfamily. We have this transforming transcription into physiology. Is that, oh, okay. Physiology and disease, and I don't need to tell all of you about the nature of the family. Uh, Basically, there's essentially 50 different receptors, as you can see in um, uh, the for the endocrine side. It's the classic steroid receptors, glucocorticoids, for sugar, mineralocorticoids, salt, and reproduction. Um, and then non-steroidal, but thyroid hormone, vitamin D, vitamin A, and these are all major metabolic pathways. And so we're very fascinated in how these pathways work together and ways in which we can take advantage of the platform with some new approaches. And and basically one just simple component here for glucocorticoids is that um, this is, this is the power behind insulin resistance. And so when you have high levels of glucocorticoids, you're going to be in the fight or flight response. And that's the first thing that comes up really high, dumps in lots of uh, uh, sugar into the body, and so that you can actually fight or flight. And you'll want the sugar to be lowered. And so there is a insulin clamp in the adipose depot. And so the uh, um, insulin resistance basically is a mechanism that is being controlled in these specific ways. And so some people think that insulin resistance is terrible. On the other hand, I think the fight or flight response is probably the most important physiologic pathway we have in our body. So I'm going to just transition to looking into sort of the next steps on what we want to do in new areas that we're going in, and in particular want to focus on FXR, which is a bile acid receptor that is what we believe is a master regulator of metabolism. In particular, it is the central driver for many different aspects of body physiology. So bile acids, and I should say that, when we isolated FXR in 1995 with Barry Foreman, he discovered it was activated by a component of the cholesterol synthetic pathway, farnesol. So we called it farnesol X receptor. And then Makishima in the Bay Area, then at the same, a little bit before us, but identified it as a bile acid receptor. And so our two teams hit that about the same time. And now, if they are bile acid receptors, just looking at this, these are the key tissues where this receptor resides. And there's about, in terms of variants of bile acids, there's at least 30 species of bile acids that we have, but with changes in diets and with the microbiome, there's quite a few additional species of bile acids that we can have. And basically, it's, principally a key regulator of lipid and glucose and energy metabolism. But some bile acids have been in clinic and have actually been approved. For example, the one that we were talking about for the obesity epidemic components, that we have the Glaxo Wellcome FXR activator, and then one that is the obeticholic acid, and that was approved, but more recently it got revoked because of the side effects and hepatic toxicity. And one thing that's important about the way that we approach the problem is a different way, and a different way of targeting FXR. And that is delivering a novel type of therapeutic. Now, this is basically cholesterol or chenodeoxycholic acid. So that would be an activator, but what I'll show you on the next slide is we made our own component of the FXR drug, which we call fexaramine or FXR amine. And this is the actual, we call it FEX, and it activates the gene networks throughout the gut, bile, and it is a major part of the presentation I'm going to talk about. And just looking down here, we're seeing that FXR is and FEX is basically only acting in the ileum. It doesn't activate any genes in the kidney, does not activate genes in the liver. But in the ileum, that's where FXR does its many different types of activities. And so there's a lot of different components here that we can sort of begin to deconstruct and to think about in terms of the different components in this pathway that get activated. And one of the things that we found, which is shown here, is it is a little bit of a surprise for some people initially, is that FXR in the gut and I should say FXD activation reduces adiposity. And you can see that in the figure over here. And what in terms of weight gain or loss, this is having actually a very specific effect. But where is the weight loss coming from? And so you want to look over here in lean versus fat mass. Almost all of the weight loss is in fat. With GLPs, much of the weight loss is in lean muscle and fat. And so, but with FEX, there's is no weight, it's really this reducing weight, but there is actually no change in the food intake of these animals. And so if you want to look at the specifics, locations of where the FXR is activating, and 51% decrease in subcutaneous fat, as you see here. So that's a pretty big impact. 72% decrease in visceral fat here, another big impact in visceral fat. Liver, a significant reduction, not massive, but and in heart, really nothing. So that's, but there's no change in food intake, even though all these components are burning energy. And so this is not one of like the GLPs that reduce appetite. These get the full appetite, they're still losing the weight and they're retaining muscle mass. And so that's actually something that is really important to think about. And so where does that fat go? And that is being reduced by the introduction of FXR into the gut. And what you can see here on this graph in terms of increased thermogenesis and consumption, we're seeing both in brown adipose tissue and white adipose tissue, direct impacts on PPRs, uncoupling protein, UCP1, DIO2. And if you look at UCP1 staining, you'll see that in the vehicle, there's not a lot of UCP1 relative to the bexaramine. So this is actually saying that we're gaining control over this specific mechanism. And so that leads to think if we can begin to manipulate that, that we might be able to tackle things like insulin resistance. And so we did look into its impact on insulin resistance, and it's really having another strong effect in glucose tolerance and ITT, insulin, improved insulin sensitivity, as you can see here. And so these two components, I think, are really very important. We can gain control, restore to some extent homeostasis, and improve crucial things, particularly like insulin sensitivity, that is very important in the global epidemic, obesity epidemic. And so thinking about the mechanisms about which gut FXR activation can increase serum things in the serum, FGF15 or FGF19 in people, these is secreted hormone from the gut. And so while the FXD does not get out of the gut, it can activate and release compounds like or gene or functions like FGF15, and that's really an important observation. You can see how strong that is, and so FGF15 reduces hepatic gluconeogenesis, even though the drug doesn't get in, and enhances energy expenditure, which is another key component here. And so this helps us. Let me get into the. Sorry about the. I go through this. Sorry about that. Sorry, guys. I got to change my computer here. All right, I just wanted to make the link between GLP-1 secretion and FXR activation. And if you look up here on the upper left, see that FEX is increasing GLP-1 secretion from murine L cells and from DIO mouse cells, and also from human L cells, which are down here. And so we have murine cells, DIO mice, and human L cells. And you can see the increases that are basically linking to what you see with GLP. And so you're seeing GLP inductions here that are really just amazing. And that's another one of the benefits, is that this program also activates in mice and in human cells the release of GLP-1. And so we have multiple aspects up here of system benefits, metabolic benefits, neurologic benefits, cardiovascular benefits now, and we're able to engage with the components that we have to really get us to another level. In the process of doing that, looking at where people are, obesity is a major risk factor for colon cancer, as you may know. Colorado is the lightest state and the lowest risk for colon cancer. Mississippi is the heaviest state. And so the question is, can we actually take the technologies we have and the observations we made to really go in and make a difference? So let's look at the impact that it might be having on colon cancer. As we can see here, just looking at the sort of a typical progression one might see in the development of colon cancer, from the normal cells to a mutation, a gene called APC or APC-min. And you get this progression here that is continuing with additional mutations to progress towards carcinomas. And we don't really understand exactly how this mechanism works. We call it the Pandora's box. Pandora was given a gift, which she was very happy for. She opened it up, and it was all these miserable things where there's sickness and misfortune and evil. And the one thing that didn't get out of the box is hope. And I think that in the way that we're thinking about it is perhaps FXR itself and FXD could be that hope in the context of Pandora's box, because what we're seeing is actually, I think, very significant, and obviously concedes many ways to think about how this can move forward. And so let's just look about how high-fat diet contributes to risk. And I should say, as this gene progresses, FXR expression is being reduced. So through inflammation and mutations, FXR is being suppressed. And in our world, I think that our goal is to try to reactivate it. And by understanding what are the initial initiations for this progression, in particular, how does high-fat diet impact on this process? And so these are some of the questions we need to know and address, which is for bile acids and inflammation. Colon cancer, there's a bunch of unresolved questions. Many people are working on them. How does high-fat diet accelerate colorectal cancer progression? How does it promote intestinal inflammation, modulate intestinal physiology, impact intestinal homeostasis, and importantly, can it be a therapeutic target in colon cancer and inflammatory IBD, bowel disease? And so that opens kind of the door to trying to begin to think about how this actually looks in real time in the body. And in this slide here from Ting, high-fat diets change the villus crypt structure. And this is the villus crypt here with healthy epithelial cells. You see it's a pretty long process, whereas on a high-fat diet, this is really reduced. It's shut down. And the arrows here, you can see the smaller arrows, are the length and direction of the intestinal epithelial cells. And the actual change in the villus space and the shortening of the epithelial cells is really, and its involution is the beginning of the real transition to colon cancer. And so we really think that we can gain control or gain purchase over what might be happening here. How does diet actually change structure? And what about, if it does, change function? And so looking at that, we can begin to look at two components here. One, and these are going to be high-fat diet in normal mice or APC-min mice. And in the APC-min mouse, on a normal diet, you get an adenoma, which you saw earlier in the diagram. In the APC-min mouse, on a high-fat diet, you now get an adenocarcinoma, which is another advanced form that you saw previously. And so basically, this shows that diet drives cancer. And that's an important thing to think about. And besides seeing the dramatic changes in the picture, it gives us, again, a way to think about how does this disruption, what is actually happening to affect gut physiology. And one of the major aspects of physiology and for bringing nutrients into the body are bile acids. And so this might be a logical way to begin to bring ourselves to think specifically, about the changes in these critical components, such as the bile acid pool. And on a normal diet, your bile acid pools are pretty low. On a high-fat diet, they more than double. In a APC-min mouse, on a normal diet, it's way beyond already the wild type. And then the APC-min alone increase, which is shown here. The bile acid pool is really high on high-fat diet. So that's, in terms of physiology, these kind of changes are major changes. So diet is really having a major effect along with the oncogene. And so that gives us multiple ways to think about how we can begin to fight the disease. And looking here at something called leaky gut or intestinal permeability assays, very little leaky gut in a wild type mouse. High-fat diet, you get some leaky gut. APC min alone, leaky gut. And APC min on high-fat diet, just look at the numbers that are different here. These are big changes, and that's a really important thing. So when we look at this and try to think about what I've told you is that when two common functions come together, we call it physiologic convergence. But when two pathologic ones come together, you get something we call pathologic convergence, like APC min, a gene, and a high-fat diet. So this is pathologic convergence, which implies that a synergy goes between those two states. And so can we disrupt that synergy? Can we take that over? And that's one way of thinking about how we might try to do that. And looking at these bile acid changes that have been very dramatic. Not only are bile acids present, but they're quite different. They come in various forms, and some are very inflammatory. And that's torabeta-muricolic acid here at the top. It increases with disease progression. It's a driver of disease progression in the normal diet. It's torabeta-muricolic acid is pretty low. On the high-fat diet, it's already jumped up significantly, and it's just not. Okay. So this is the graph that I was showing you. And then in the APC min, this was the wild type on the left. On the APC min mouse, you can see normal diet, high-fat diet, there's actually a 50-fold change in torabeta-muricolic acid. And that is crazy. But this is what is driving, we believe, tumor progression with the APC min mutation. And these kind of changes can occur in many different forms, but we think now this might fit well into the way we are thinking of how to sort of visualize this with disruption of homeostasis that is kind of shown here from hereditary things and the environment, which is food, and then unknown things about mutations. For the genetics, we see the mutation here with torabeta and the high-fat diet here that generating this torabeta-muricolic acid and deoxycholic acid. And so there is a direct targeting of all of these drivers to FXR. They want to suppress FXR. They're designed to suppress FXR. So these dietary risks in this cascade of events that we see coming is able to give us a purchase on potentially reactivating FXR signaling. And if we can do that in the context of all of these confluent and mutation in inflammatory drivers, can we do anything with it? And so we're going to look at that in a little bit more detail. So one of the things in this context, again with the wild type and the APC min, is looking up here in terms of normal diet. This is back to the intestinal permeability assay. The intestines tight and clean in the wild type space on a normal diet. With APC min, you can see it really goes up. But now we can use our FXR drug, FXD, and look what it does. It brings this down from 150 to below 50 with the drug. And so that means the drug is overtaking and overcoming some of the other parts of this, such as the adenoma or the high-fat diet effects. We have to really break that down in more detail. In the adenocarcinoma, where it's even higher, what you see here is, again, the wild type is relatively low in this, and in the high-fat diet component, what you see is, again, that the mouse that is receiving FXD is really lowering the leaky gut component, which is really amazing in all these different models. And so FXD restores gut integrity in both adenomas and carcinoma models. And so there's another couple ones I think that you'll find interesting to see is how additional impacts on the bile acid pool in slightly different way. And again, what we're looking at is this major change of bile acids, which are fundamental. There are primary and secondary bile acids as well, and pretty quiet on this part of the normal diet. But if you put them on a high-fat diet, which which is over here, or give the drug here or here, you see that no matter what, whenever something goes up, we can get FXD to bring it back down in both the adenoma and the adenocarcinoma. And so this gives us a lot of, I think, real belief that while these, there's the confluence between the negative and inflammatory regulators that is not impenetrable. And so looks like we have ways to maybe advance this to the next level. And so let's think about how we might want to do that. One of the ways that we've been working on this is looking at survival. And if you look at FXD, it increases the lifespan of the mice here, and at least by 20 weeks. And these are actually hard to get extensions in the APC-min model, and also the APC-min model on high-fat diets, which it does also extend, but it's shorter because it's such an aggressive impact of it. And so what we see here in looking through this is there is something that can extend life, number one. And number two is that we might be able to use the gene expression changes that are coming in from normal or vehicle to FXD. And so with these DNA genetic changes, we can see both not just the increased survival in both the adenoma and carcinoma, but we can actually take this to a human database and run that data set against the human colorectal cancer patients, and you can see that the ones that have the FXR FXD component is really showing a longer lifespan. So if you have the FXD activated network coming, that you're going to have a lower risk. And so it looks like it could potentially extend to people. And that's actually looking at organoids from mice and from humans as well. And so I think all that is really interesting, leading to a short summary of bile acids, which are early sensors of dietary fat. There are also genetic and dietary risk factors for colorectal cancer. And we want to target this is BA and FXR axis. This is the pathologic convergence, leading us to leading to tumor progression and to intervene in all these malignant phenotypes. So this is a little bit like where we can see more in Pandora's box with pathologic bile acids as well as part of that's in that box. And they also promote tumorigenesis. But we think we can reverse many of these components and concurrently that can really have major impacts on colon cancer, its impacts on LGR5, and ISCs are really, I think, pretty effective. And the last component here is that FXR agonists really, in our data, restore gut health, and they curtail tumor progression. That's big. And because of that, this opens a door, we think, to a new era in tackling one of the most challenging cancers. And so this is one of the core areas that I think we can really advance. And just looking generally at 4 components to look at is metabolic disease, which is really part of the program that you guys are running in obesity and diabetes, because affects, it triggers the GLPs, it burns fat, it improves insulin resistance. And in the gut, colon cancer, it does have positive benefits with inflammatory bowel disease, Crohn's, and ulcerative colitis. So again, it is reducing the risk in the risk factors. And in the liver, even though FXD doesn't get in to the liver, it has a direct impact on NASH, fibrosis, liver fibrosis, cirrhosis, PBCs, and PSCs. And so all these are leading to reductions of
What's up? Your phone's ringing.
Okay, guys, my phone is ringing. Sorry about that. And then the fourth one is on additional organs. And so besides just the gut, we actually are looking at the pancreatitis, which is a really challenging disease to treat. And chronic kidney disease, CKD, also not an easy one to treat. And also, many people have to just get kidney transplants. And there's components that have central activity as well. So from this one molecule and many uses. And so the genetic and pharmacologic experiments here to combine to the direct development of intestinal targeted therapy is really the goal. And I think it's one that is very possible to have. And I'm trying to see if I can say, I think I'll just go here. And here we go. So I just want to thank everybody. Ting, still going great. And a lot of other wonderful people in the lab that help to move these along with our collaborators over here, mostly at UCSD, but actually quite a number of different locations. And really so happy to be here, talk to you guys, think about the next steps, and how we can actually move this entire platform forward. And so I guess if you want to go into questions, I'm here.
All right. Thank you. Thank you so much, Dr. Evans, for the beautiful work. I'm the co-hoster of your today's talk. So I think we learned a lot about the bile acid and the leukoreceptor receptor function today. We're now moving to the QA session from our panelists. So first, Dr. Wendell Huang, please go ahead.
Hi, Ron.
Hey.
Nice talk. I always enjoy your seminar. And also, I'm very happy, among so many nuclear receptors, you talk about FXR, because when I first joined Dr. David Moore's lab at Baylor, I start with CAR PXR, but I was very happy, I was able to switch to FXR.
I know. I remember all that.
FXR is more fascinating.
Yeah.
And okay, so my question, my question for you is that you know FXR initially was developed to treat NASH by Intercept, but it failed approval from FDA, and then another receptor, thyroid hormone beta receptor agonist, get approved. The drug resmetirom, so it's now it's clinically used to treat NASH. I hope you can. Do we have any comments on this? Is thyroid hormone beta is the best for the agonist is the best for to treat NASH?
For right now, yes. There are new ones coming along.
Right, because there are also PBAR under clinical trial.
Those are coming along.
They are going to get approval also.
Yeah, exactly. And hopefully they'll work, but this is working in a different way, in a different fashion. And in the context of these diseases, I think the combinations could be very potent. And you can see with FEX-D, it doesn't get in, and it's not toxic anyway. And so you're getting the benefit of something that's able to have what you might call a resolution driver in terms of the cancer to reset the program, its natural program. And so that just gives clinicians lots of opportunity.
Okay, thanks. My second question is about this fascinating also the FXR, the gall restrict FXR agonist that fat. So because they are strong phenotype in the adipose tissue.
Yes.
So do you think this could be a secondary effect by activating the TGR5? TGR5 is highly expressed in the broad adipose tissue.
Yeah. I mean, that's a good question. We actually haven't tried to dissect that. I mean, it is a very good question because we do want to know how it is working. But the one thing is these are things that you can actually break down and figure it out. I think we're pretty close to that as well. So, but you guys also have a strong program in this space.
Yeah. Okay. So I can keep asking you questions, but I like to leave that time for other panelists and other people who are interested, who will ask you more questions. Thank you, Ron.
Yeah, of course.
Thank you. Next, Hong Ying Gao. Please go ahead.
Hi, Ron. Always fascinated about your talk.
Thank you.
So my question is that you mentioned about during the colon cancer progression that FXR, is that level decrease or is this the activity decrease? So if it is a level decrease, what control, what is the mechanism that FXR
For which
causes
one?
level
Which one are you
decrease?
talking about?
FXR, when the colon cancer progression, right?
Oh
So
yeah.
you talk
Okay.
about
Yeah.
FXR, its expression decrease,
Yes.
correct?
Yes.
Okay. So what controls the expression? And the next question is that you mentioned about diet, diet can change. What about the behavior, such as, exercise or smoking or drinking? Would that, make the disease worse or maybe improve the symptom?
Oh yeah, but yeah. I mean, could giving Fex to someone who's either diabetes or um, did you?
So the question is, does exercise increase FXR?
Okay, first one, exercise. I mean, I don't think we know that. It's a really good question, but we do know that exercise does have a beneficial effect in general on uh, um, uh, cancer in general. And the studies have been done
Yeah.
so that, and the reality is we are, as animals, we're designed to move. And so if you don't move, because, we don't have to like hunt and do all that stuff, and also with technology, we are less, um, we move less because we're at our computers and our phones. And so I think that is part of the epidemic that is causing increased weight gain. And I think that we need to think about ways in which to really reel that back. And you just can't just only do it with regular diets with the GLPs. It's going to be better, but I think that we have an advantage with a drug that doesn't even get in the body, and it has all these multiple simultaneous effects, and so that it is having the impact concurrently that further, I think, makes that kind of therapy better. And we have to get this to the next level, but I think that there's a lot of I think that we're going to see a lot of new programs in the near future.
So you mentioned about there's some research about microbiome. Maybe they have some metabolites, have some bile acid also function as a FXR ligand. So can you elaborate with that?
Yeah, I mean, so the bile acid pooling, and again, this is one that the Ting has been part of as well, is that the bile acid the bile acid pool is really pretty heterogeneous. There's a lot of different components to the um, uh, and which depend a lot on what you eat. So the microbiome adapts to what you eat, and they evolve their chemical composition as well that follows the kind of things that you're eating, and they also are generally inflammatory. But I think that the main thing for kind of the next stage of how we might think of this, and I think that you're so your question, I think, is really important to try to figure out. But I believe that if we can get get sort of to the next level, where we can actually sort of deconstruct these components that with diet, we almost have to have a way to take advantage of the physiologic networks that we have, and to use those to sort of engage and promote health. And so an FXR, I think, will be one of those. But and I think that, as you mentioned, there's other programs for trying to improve liver disease. I do think that we're pretty close to some breakthroughs.
Thank you.
My follow-up Hunying's question about exercise. Ron, you also have the PPAR delta agonist. That's kind of mimic exercise. Do you think the PPAR delta agonist have synergistic effect with your new FXR intestine-specific FXR agonist?
I mean, can they be used together?
Yeah.
That's a very good question. And we did actually finish a trial with one of our PPAR delta compounds, and was successful. It was actually had an impact, a beneficial impact in the heart. So I would like to do that experiment. I think that PPAR delta activates oxidative programs that are linked to motion, and they burn fat and they restore integrity in many organs and tissues, including the heart. And also, they probably have some central effects as well. Because anything that is promoting exercise, basically, is going to activate the networks, the gene networks, that are going to really restore health. And so that's all doable. I think it's kind of the next wave, I'd say. And combinations are always going to be, I think, interesting if they can be done correctly. You don't want to overdo it, but I think that there's going to be lots of opportunity in this space.
Thank you.
Okay, next, Dr. Xiaoyong Yang.
Yeah.
Hey, Ron. Yeah, you see you, Ron. So it's always a crystal clear presentation. And one question is like, you show like there's the FAXD, which is specifically active SR in testing, that can induce a secretion of GLP as well as the FGF19. So like a side effect of GLP is a muscle loss.
Yes,
which is right. In the FXD, which means GLP is not a major mediator of beneficial effect of FXD. Is that true or no?
Well, so the FXD does, with FXD you get weight loss, but it's not in muscle. It's not in muscle. It's only in fat. Right. And there's no change in diet either. So they're not losing it because they're not eating. They eat the same amount. And so this is very different than the GLPs, right?
Mm-hmm.
And so, but of course, part of the FXD is also activating the GLP network, but it's activating it in a different way, where GLP is only one component. When you take GLP, you got whatever that is there. But the way we do it, it's a diversity of genes that are part of the FXR network, and that overcomes, I think, many of the inflammatory and disease components that I think give the sort of non-absorbed drug like XD, I think gives it an advantage.
Yeah, what do you think any potential side effects of XD we know about?
What of any what? Say,
Side effects, the side effect affects the any potential.
it is actually it has very good effects just in ting-nosis as well, but in the gut, it's having a great effect in terms of the microbiome, because that was raised a little bit. Typically, people who are on high-fat diets have inflammatory, has a different set of compositions of microbes. And when you give FEX-D, and they stay on that diet, but when you give FEX-D, it totally reprograms the microbiomes. And so it gives you a different microbiome signature. And so it is also gaining control over that major entity, which is the microbiome, and restoring and getting sort of suppressing the inflammatory components and enabling the healthy components of the microbiome to be functional. And so it has a really interesting effect that, again, is the benefit of having the way in which the drug was structured.
Yes, thank you, Ron.
Yeah.
Just following that question, so I wonder, have you looked at the fat absorption?
Have we looked at fat absorption?
Right.
I'm not sure what you mean by that, though. And what we
Well,
do.
for the gut specific FXR activation, I mean, in that model, because you showed there's no difference in food intake, right? But it has some effect for the adipocytes.
Yes, yeah, it does.
Oh,
Which is,
for what?
yeah, so that's a good point. We can't say exactly how that's working. But I'm glad you asked, but I can't give you unless Ting can do it. But I think that it is what to be able to lose that without losing muscle mass, I think, is the big thing. And but I'm not sure exactly in sort of what
Yeah.
I don't think I can answer your question yet.
I think we measured the triglyceride, at least the triglyceride is also reduced by the FASN D.
Yeah.
So yeah, the fatty acid, triglyceride, but we didn't measure the absorption just like in intestine to do some in vitro study. Still like phenotypically all the in vivo study, we saw that too. Yeah.
Yeah.
And we actually saw that more fat is going to be triglyceride, going to be into the feces. It's yeah, it's interesting.
Yeah, that's interesting.
Yes, you're right. You put it out.
Yeah.
That's interesting. Okay. Next panelist, Dr. Yin Lei. Yin, are you here?
You're muted. You're muted.
Yeah,
Okay.
thank you for the wonderful talk about FXR. I haven't followed FXR for a long time. I always wondering is there any genetic component in terms of within the FXR pathway that leads to the sensitivity to various metabolic disease? For example, in patient progression from the fatty liver simple steatosis to steatohepatitis. Has anybody ever done any genetic study to show the people progress to that, the more advanced, fatty liver disease has a genetic mutation in terms of the FXR pathway?
Um, I don't know that we've completed an analysis. Then we did begin to do that, but I don't think that we know much more. I mean, I know what you're trying to get to, but and it's a little bit surprising how many things FXD is able to do by just going through the gut. And but yeah, we don't have all. I mean, that's partly what I think is the fun part of what we're doing, is deconstructing how this actually works. That gives us both better insight and better control over the kind of epidemic that we have. And so I think that there's a lot of questions to address, but I think we've made good progress.
I'll just follow up those questions. So is FXR pathway, when it's impaired, is the primary cause for dye-induced MASH, or is somehow in the middle, like a secondary, tertiary?
I don't know that the pathway is turned off. I mean, that's what you're asking, right?
Yes, yeah.
And so I mean, I think it probably is to some extent, because you know, because we're not talking about FXD, we're talking about FXR in the gut that is being either activated by some of the bile acids that are there naturally, that's going through, and so or being sort of suppressed by inflammatory components, like, you know, lipids that are inflammatory, and so different ways to think about you know, what the part of that program is. I mean, there are a lot of programs in the gut. I think that's one of the things that probably we don't know all of them. What we're seeing, there's a lot that goes on there in different parts anatomically. So, but basically, in an environment where there's high fat diets consistently, I think that's where the challenges in general for the dramatic changes in microbiome and dramatic changes in what become inflammatory components, and then you get inflammatory bile acids. So I really think that if the weight loss is in mice, if it's as good with FXD as it is in people, then I think we have a way to look into this in a little bit deeper way. But I think that, I mean, it's a good question. I'm not sure I could give you a good answer.
Thank you.
May I follow this? They ask genetic. I'm wondering, you know, female and male respond differently to heifer diet. So in this FXR agonist, will the female respond better compared with the male?
That question is so easy that I think Ting can answer that.
Okay.
I think the ER can also regulate the bile acid as well. So I think always like in the male, it's actually more consistent. Yeah. So I think for like a Dr. Lay's question, so I think FSR has like some polymorphism like in the colon cancer. And also there is some report about like FSR like promoters are going to be methylation. So that's another, in some tumors, so that's another reason why the FSR expression was downregulated in the colon cancer patients. Yeah.
Yeah. You don't want it to be downregulated, but it needs to be downregulated for the cancers to progress.
Yeah. I have a question about like the GLP-1. Because there was some like a study about like the GLP-1, like FSR inhibition was like a induction of the GLP-1. But it seems like there was always like it's like a pro glucagon level or it's like a secretion level. So what it's a direct or indirect. So what are your thoughts about it? Like, and also because the result is like use the cell line, some people use it in vivo. So I was quite confused about this part too. Do you have any like insight on the, how to elucidate like this question?
Well, I mean, it is
Right.
yeah. I mean, it is a good question, and but again, that's not an easy one to answer because the GLPs are uh, that's kind of a sort of independent mechanism that's going on in the body, but it turns out for reasons that no one really knew, that there's a lot of GLP that naturally comes from the gut, and it comes from the gut in the context of FXR being activated. But if you have high fat diets, you're not going to activate FXR, and then so there will definitely be changes in GLP depending on the foods you eat and other things that, like we talked about, exercise as well. So I mean, it's a good thing, but you have to run the trials like probably on treadmills or really specific diets where you can actually get the information you need. I think it's a good question.
Okay, Dr. Evans, we also have some questions from audience. Would you mind if I read for you?
Okay.
Okay. So Zhou Huali was asking how a non-absorbable FX activates FXR in the nucleus.
Well, that's how it was designed when we did it here, and so we really we started with a library of about 100,000 or about that of compounds that we were running for our assays, and then and we found a couple different ways doing crystal structure to evolve the platform, and so there's a little bit of luck in that, but our goal there, which I've kind of said before, is by making it intestinal restricted, you gain a lot of impact, keeping in mind, because we've already showed in our own data that FXR receptor is present throughout the gut, and so it is. So this tells you that it's a major target for our body physiology, and so I think that it's a little bit hard to say about with some of the different diet components and other things like that, or the way in which people are trying to lose weight. I'm not sure how it impacts the FXR network. I mean, it's a good question, and again, to go back to microbiome, there might be ways in which the microbiome can modify it. We know it can generally modify diets, but we also know that FXR is very potent in shutting that process down. So, I mean, these are all interesting areas. I mean, you think you know everything about what's happening in the gut, but you don't know it all. But I think that there's a lot of reason to believe that there is a good way forward.
The next question is there any difference between intestinal FXR activation and systemic FXR activation in the regulation of fat metabolism?
Yeah, I mean, that is a good question, and that's where almost everyone in the pharmaceutical FXR drugs that were systemic. And at least some of those looked promising, but they didn't really gain traction. And we were aware of that, and that's part of the reason for trying to make one that is not systemic, because see, the FXR is not evenly distributed through FXR receptors are not distributed equally throughout the body, as you saw kind of in the map that showed you at the beginning, and so it's a little, and we've looked at oral FXR therapies as well, and I think they could have a chance to do it. And there are companies that are actually doing that, but I do think the future is not ignoring what's going on, but actually at least evolving the FXR platform to be able to sort of strengthen the whole environment. It seems to me that there's a lot of good things going on, and the GLPs are kind of they do the 4 components that they kind of get activated in, which are very important, and so yeah, I mean, and there might even be a future of GLPs with effects component, although it makes so much GLP, it may not really be beneficial.
Question from Wits Zhang. Had the FAXD been tested in PBC mouse model? If yes, which model has been used?
Ting, I don't know that I don't think we did that.
No, we didn't do that. Yeah.
And
I think the PBC model probably pretty hard to establish, right? I think the PBC model, some people do the GFP, TGF beta receptor, like dominant and active one. Yeah. So they can mimic the human PBC. We didn't try that model, yeah.
Yeah.
Move to the next one. Zhiqiang Huang was asking how to understand the role of the nuclear receptor and non-nuclear receptor function in the context of bile acid activation of FXR.
Well, wait, say that again. I'm sort
of. Sure. How to understand the role of the nuclear receptor and non-nuclear receptor function in the context of bile acid activation of FXR.
Well, okay, I'm not quite sure exactly all that is being asked there, but there are, besides FXR as a bile acid receptor, there are at least 4 other receptors in the gut that are also bile acid receptors. And so when you get the first one, you think that's it. But there now are, and we know, are a multitude of different forms of bile acid receptors, and also there's different forms of bile acids. And so the nuclear receptor superfamily is going to be a target in general for those kind of potential targets. And truth be told, we haven't looked at, once we had FXR, we didn't go too deep into some of the other parts of the nuclear receptor family that might also respond to a bile acid. Although I think a lot of it's been done, but yeah, in general, there are more bile acid receptors than we thought. The dominant one is FXR.
That's a big question. Yeah. Question from Hui Han. In human, reduction in FXR activity in obesity is also because of the the taural beta
Yes.
uracolic acid, yeah, or other mechanism.
I'm sorry again. Just say it again.
So reduction in FXR activity
Yeah.
in obesity. I
Yeah.
think this is for
Yes.
the first part of your talk today.
Yeah. Yeah.
Is that also because of the taurate beta muric acid or
Oh, yeah.
some other mechanism?
Good question. I would say that it's a good question. It probably that is part of that because uh, for the progression in the gut or other tissues, but let's say in the gut, like we were talking about, um, that is it's oncogenes are going to want to get rid of FXR. It's the same thing is also true for like the vitamin D receptor. There are enzymes um, in cancer cells that specifically break down vitamin D because vitamin D is anti-inflammatory. And so tumors know how to get rid of that. And that may also be true for the FXR drugs. The thing is that the RFXR drugs, because they're synthetic, there's no natural mechanism that would target them. So that may be an advantage to override some of these other components. And for regard to other nuclear receptors, I don't think there's been I mean, it's a good question. It'd be really nice to look at all of them in a thorough way, but it's a big effort. And but so far, I don't think there's much to say at that stage.
The last two questions is regarding GLP-1. So I think Dr. Evans, you already answered the first one. The last one is about the GLP-1 transcriptional activation. So Jie Mom was asking, is if FAXD does FAXD promote GLP-1 secretion through transcriptional activation mediated by FXR? Because she didn't see any MRI data for the GLP-1 in your slides. So she was wondering if you find that FAXD acts through other pathway to enhance GLP-1 secretion.
How is it? Yeah. I'm not sure we can say all of that. I mean, what we can do and we have done is we've looked at the induction or activation or the release of GLP-1 in some different contexts in the gut. And but not sure if I'm not sure that we again know enough to if other if there are other mechanisms that either enhance it or actually antagonize it. I mean, it's probably not the I'm not sure if that's the right answer for that question for FXR, because we haven't really done much there.
No, any more question?
I think that's all. Is kind of late. Yeah.
So
Okay, guys.
A lot of questions.
No more questions.
Yeah, maybe one of the longest Q&A recently for Ron.
Yeah.
But thanks a lot, Ron. Yeah,
Okay,
we have like 410 people's audience.
Oh, awesome.
Yeah, a lot of
Super
people.
nice.
Thank you so much again for joining us today. Thank you, everyone. Yeah, I hope you all enjoy